Hello, my name is Dr. Roderick L. Rowe, and today I will be talking to you about physical methods of controlling microorganisms. Sterilization destroys all microbes and viruses in or on an object. The only exceptions are prions, which are proteinaceous entities that cannot be destroyed, and hyperthermophiles that cannot be destroyed. The prions, you probably talked about it in your general biology class. There was this classic example of the Kuru people who ate each other. They were cannibals. And this disease was the Kuru disease, and it was disseminated when these individuals ate each other. So what they discovered is that the prions are in the nervous tissue. And because it's in the nervous tissue, individuals develop a disease. So if you eat a person and you ate part of their nervous system, you therefore got the disease. So prions cannot be destroyed. They're just proteins. So when we start talking about destroying organisms, we're destroying a cell that has a nucleus, organelles that have function. If the microbe is just a protein, then how can you destroy it? So, sterilization destroys all microbes and viruses. Microbes are cells, viruses are not cells. Disinfection is the destruction of most microbes and viruses on non-living surfaces. So the action is disinfection. The thing that is doing the action is the disinfectant. And the work is the action, the actual destruction. So in lab, you're going to use conflict every day. Conflict is a disinfectant. It is a chemical that has some type of entity, some type of compound that will destroy microorganisms. And that compound is usually at a higher concentration than something such as an antiseptic. So an antiseptic is a lower concentration of a disinfectant. So the action is antisepsis, and antisepsis consists of a lower concentration of the disinfectant. So let's say the disinfectant used 10% quaternary ammonium compounds. If it used 10%, then antiseptic can use 2%. So the antiseptic is going to destroy microbes that are on living surfaces, such as your skin. So the thing that is doing the work is the antiseptic, and the work is the destruction. If a pathogenic microbe gets into blood or tissue, the patient has sepsis. Now you should have a better understanding of what antisepsis is. Sepsis is when the microbes are inside or on your tissue. Antisepsis, we're destroying those microbes. Remember that you have to use a lower concentration of the disinfectant. We're going to look at some prefixes and suffixes throughout this chapter. Right now, let's just look at the suffix. The suffix CIDE, side. Whenever you see side, something is being destroyed. So bactericide is bacteria that are being destroyed through some type of disinfectant. Fungicide, we're killing fungus. Germicide, we're killing a wide range of microbes. Whereas if you look at this other suffix, static, This means to stop the growth. The organism is not dead, it's just hindered. So bacteriostatic means that we're somehow slowing the growth, stopping the growth of this microorganism. Maybe the refrigerator is doing that because of the cold environment. It is restricting the microbe from dividing. A disinfectant is used to create an environment free of microbes. So in lab, you're going to constantly uh, spray conflict onto your table surface. You're going to constantly put on gloves and spray conflict on gloves. You're going to spray ethanol on gloves. You're going to wash the gloves constantly because you're trying to create an environment that is free of microbes. An environment that is free is called aseptic. So in lab, you're going to transfer organisms from one solution to another solution. This is called aseptic transfer. The most appropriate way to do this is, as you can see in this picture, we have these chambers that you can stick your hand into the chamber. Your hands are in gloves, and then you can manipulate whatever the product is that is inside. Contamination is when disease-causing microbes are present. Contamination, decontamination is when we remove those microbes. And we can remove those microbes by destroying them by using things such as disinfectants. Sanitization is the removal of pathogens to meet public health standards. This can be achieved by either cleaning or sterilizing. In most public bathrooms, they just do cleaning. They just clean the surface to make it pretty, but it's not necessarily sterile. Your bathrooms are probably cleaner than most public bathrooms because you use strong, high concentration disinfectants. These janitorial service companies are trying to make a profit so they can water down their disinfectant. Funny story, I used to work at a gym in Tallahassee. It was a university gym called, it was at FAMU. And you've probably seen Simple Green before. It is actually a green disinfectant. So I caught them bringing out squirt bottles or spray bottles that had a very, very, very light green solution in it. And I said, well, what is this? And they said, well, that's Simple Green. Well, green is a emerald green. And this was a very, very light green. So I called them out on it. So trust me when I say, for public use, it's not really clean to how I would want it to be cleaned. It's going to be a diluted concentration of a disinfectant, of some kind of compound that is supposed to kill. Now in lab, when you read conflict, it told you that that concentration needed to be on the surface for 5 minutes to kill everything. So if we dilute it by 50-50, water to conflict, then it's no longer killing the microbes in 5 minutes. It might take 30 minutes to do the same thing. So if any of you ever work in a janitorial business, please don't dilute the disinfectants. Degerming. Degerming is the removal of microbes by mechanical means. So using bar soap is very effective at removing microbes. Notice I didn't say destroying microbes. A disinfectant will destroy a microbe, whereas a bar soap will not destroy the microbe. It is going to package the microbe into these individual micelles. So that micelle is a fat molecule. Well, there's many fat molecules associated with it, and the little microbe is going to be inside of the circle. So when you wash hands and you lather, that is what you are doing. You are packaging the microbes into these neat little packets of fat molecules called micelles. So I have a question here. Why is antibacterial soap not good for hands? Which is healthier, bar soap or antibacterial soap? So think about not just the killing capability or the removal of the microbe, but think about the harsh chemical that is on the surface of your skin when you answer this question. Which is healthier, bar soap or antibacterial soap? The CDC has this information about hand washing. Notice if you read this box here, it says studies have not found any added health benefit from using soaps containing antibacterial ingredients when compared to plain soap, the mean bar soap. Both are equally effective in getting rid of germs. So in reference to getting rid of germs, they're both equally effective. But notice that the antibacterial contains at least 60% alcohol. Now think about what alcohol does to the surface of your skin. Now if it's an emergency and you went out to eat and you use this antibacterial soap, well then that's okay. But what if you are a nurse, and nurses may wash their hands 100 to 200, 300 times a day, and you're washing your hands with 60% ethanol. Remember, ethanol, isn't that a disinfectant? Yes. At a high concentration, it is a disinfectant. At a low concentration, it is an antiseptic. So with that high concentration, it is not good for the skin. So over to the right, I have a picture of the skin. The skin is the integumentary system. It protects the body from penetration of microbes. So microbes have to somehow navigate through all of these layers, the stratum corneum, the stratum granulosum, all these different layers the bacteria has to get through, which it cannot. But if you use 60% ethanol 200 times a day, you're going to start having these micro fractures, these cracks all throughout your integumentary system. And those cracks are going to now be able to allow bacteria to come in. So be mindful of that. Yes, bar soap and antibacterial soap are both as effective at removing the germ, but one of them is not good for your skin. So just be mindful of that. We're going to talk about physical methods of controlling microorganisms. These physical methods can be divided into 7 different categories. The first one is extreme heat. Then we will discuss extreme cold, followed by desiccation. The fourth is lyophilization. Then we'll talk about filtration, exclusion. Then we'll talk about osmotic pressure. And then we'll end with radiation. So what we will conclude this slideshow with is just talking about the heat. So we got different types of temperature, different types of heat. The first one we'll look at is moist heat. Moist heat is used to disinfect, sanitize, and sterilize. Remember, sanitize is just for public use. Sterilize is what you want, where it kills everything. But moist heat can be used for all. Moist heat kills by denaturing proteins and destroying cytoplasmic membrane. Remember, the structural protein has, let's just talk about the globular protein that has a 3-dimensional shape. And in order for it to be functional, it has to be into its tertiary or quaternary structural formula or format. Remember, hydrogen bonds hold the tertiary structure together, along with other bonds. But the hydrogen bond is very, very weak. When you start increasing the temperature, you start to break those hydrogen bonds, then the proteins will unravel. You talked about that in general biology, and it was called denaturing. So if you denature these proteins, and these proteins are enzymes that are involved in the day-to-day living of the microorganism will die. So that is how heat will destroy the microorganism. So moist heat is more effective than dry heat. Why? Because water is a better conductor of heat than air. If you had to choose option A, stick your hand in that oven at 400 degrees Fahrenheit, or stick your hand over that boiling water at 212 degrees Fahrenheit, which one would you do? You know which one you would do. You would stick your hand in the oven, and it's not that hot because it's dry heat. Now, if you left it in there, it's very hot. But if you stuck your hand over that boiling water right when you open that pot, you're going to burn the hair off your hand. So moist heat is a better conductor of heat than air. 5 methods of microbial control use moist heat: boiling, autoclaving, pasteurization, ultra high temperature pasteurization, and ultra high temperature sterilization. Boiling kills vegetative cells of bacteria, fungi, protozoans, and these protozoans have different stages of life. The feeding stage is called a trophozoite. The dormant stage is a cyst. So boiling kills the trophozoites and most viruses within 10 minutes at sea level. So whenever you have a boil water alert, some sewage leaked into the drinking water supply, you don't want to pour a glass of water and there's feces in it. So they tell you to boil the water. Well, how long should you boil it? At least 10 minutes. Boiling water cannot exceed 100 degrees Celsius at sea level. And because that steam is going to carry away heat and energy, if we could somehow get that temperature above 100, then boiling is even more effective. But we cannot get the temperature above 100. Why? Because the steam is carrying away the energy. So how can we get this temperature up? We have to put a lid on the pot. Put a lid on it, you're going to start to generate pressure. But you'll be able to get that temperature above 100. Endospores, protozoan cysts, and some viruses can survive boiling for many minutes to even hours. Some bacterial endospores can survive for over 20 hours. Even thermophiles can also survive. So is heat very effective at killing? Yes. Is moist heat effective at killing? It is absolutely effective. But does it kill everything? No. Moist heat cannot kill everything. Look how long you have to boil just to kill endospores that comes with bacteria like Bacillus thuringiensis or Clostridium tetani. Bacillus and Clostridium are both resistant to boiling because of their endospore. Endospore is nothing more than sort of like an egg. So the bacteria can produce a dormant cell. This dormant cell is an endospore. And if it's too hot, if you're boiling it, it just stays protected in the endospore. Then as soon as it cools down, as soon as sugars are present, then it will germinate and turn into a vegetative cell called bacillus or Clostridium. So boiling is only good for sanitization or sanitizing restaurant, tableware, and baby bottles. Autoclaving is better at sterilizing because it is going to utilize pressure. So just like the pot that was boiling and it was losing a lot of steam, if you could somehow seal that, if you could somehow seal that same setup, you would be able to increase the temperature from 100 to maybe 121. And just that little bit of increase in temperature will now be able to destroy all of those organisms that we just talked about, even the endospores. So this apparatus is called an autoclave. In this picture, you're looking at one. It has this big metal door, and I showed you one in class. You're going to close it up, you're going to heat it up, and it's going to kill everything. Remember, you have to put a tray inside of this autoclave because if not, if agar is present and it boils out, it can get into that, see where it says steam supply? Some of your agar can fall into that little pipe or this other one that says air. And if it gets into these valves, then it will destroy or clog this $50,000 machine. So here are some of those huge autoclaves. My brother worked at he's a mechanical engineer, and he worked at a facility in Daytona where they create the saline bags. I discussed this extensively in general biology. Well, this big chamber, they can put entire crates of saline bags, entire crates of saline that need to be sterilized. Just make sure you don't get caught inside by yourself. The boiling temperature increases as pressure increases. So once this container is sealed in this bottom picture, which is a pressure cooker, once it is concealed or sealed, the temperature is going to increase as pressure increase. So autoclave conditions usually are 131 degrees Celsius, and you're going to generate 15 psi, and we usually cook it about 15 minutes, and that typically destroys everything. We're going to skip the coloring activity. Let's talk about pasteurization. Pasteurization is a heating method that does not use pressure. The temperature is raised a little, which does not ruin the taste of the solution. You don't want your milk to taste different, so we therefore pasteurize it. So pasteurization is used for milk, ice cream, yogurts, and fruit juices. Pasteurization is used to destroy pathogens. It is not sterilization because heat tolerant and heat loving microbes will survive. You don't get the temperature that hot with pasteurization. These do not cause spoilage, the microbes that are left over prior to consumption. These are generally non-pathogenic. You notice I say generally. Well, I always purchase the same milk from the same company. Some of y'all like to go and find deals. Just be careful when you find this random manufacturer of a milk product. They might use a different form of pasteurization. Their temperature might be a little different. They might not kill all pathogenic organisms. So just be mindful of that when you drink your milks or purchase your milk. The holding method is one type of pasteurization, the holding method. It is also called the batch method. It is an older method, and it heats the solution up to 60 to 62 degrees Celsius for 30 minutes. It is less likely to change the taste and the nutritional content of the solution. It is also more effective against resistant pathogens such as this one that causes Q fever, Coxiella, and Mycobacterium. So pasteurization is a good thing because it kills a lot of these microbes that need to be destroyed. So that was the longer time frame method. If you notice this one, the temperature is a little bit higher, but instead of 30 minutes, now it's only 15 seconds. So this is called flash method. It is a newer pasteurization method that doesn't heat the solution as long. Both the holding method, batch, or flash method inactivates 99% of bacteria, fungus, and viruses. Endospore and heat-resistant microbes remain. Some of these heat-resistant microbes are lactobacillus, mycococcus, and yeast. These organisms cause the milk to spoil. So if you could heat it longer and destroy lactobacillus, mycococcus, and yeast, then the milk will not spoil. So if you see milk at Costco and it's just sitting out on the shelf, it's probably been sterilized or probably was heated up enough to kill all microbes that could spoil the milk. That's why it has a different shelf life. Sacrotropic organisms are cold-loving organisms. Some of these organisms can exist in milk, and listed below I have a few examples. So milk is typically put in the refrigerator. Well, do organisms still live in the milk when it's in the refrigerator? Yes, some organisms can tolerate this cold environment, and they are sacrotrophic organisms. The ultra high temperature pasteurization method might affect taste because it heats up twice as high as the batch method. It heats the solution up to 134 degrees Celsius for 1 second. There's another method that also uses the same temperature. It is ultra temperature sterilization. So instead of 1 second, we're going to go to 3 seconds. So this is going to kill everything. So this sterilization method is used for dairy creamers that are served at restaurants. It will chemically degrade the sugars and the lipids and the proteins that are inside of milk, that are normally inside of milk. It's definitely going to chemically change those molecules. But sterilization is going to kill all living microbes. Then storage at room temperature is possible without spoilage. So long-term storage can alter the taste. This table here is a summary of all the different methods I just discussed. Listed below are some milk-borne diseases caused by the lack of pasteurization. Why did I put this slide here? Because everybody's into living healthy and trying to be organic. And I've heard students talk about they don't want pasteurized milk. They want their milk to be unaltered. And I'm telling you that these are some of the organisms that you might be exposed to if you drink milk that has not been processed properly. So the milk needs to be pasteurized. So you can get salmonella, that's a food infection. You can get this Campylobacter, listeria, brucella. You can get all these different infections because your milk is unpasteurized. So that was moist heat, and now we have dry heat. So dry heat from an oven can achieve sterilization also, but you'll have to heat it much longer than what we just discussed. We talked about 1 minute, 1 second, 2 second, 30 minutes, 15 seconds. Now for dry heat, you have to, at 170 degrees Celsius, you have to leave your item in the oven for 2 hours. So dry heat is used for materials that cannot be sterilized with moisture. The heat denatures proteins and oxidizes metabolic and structural chemicals. So that is how it destroys the microbe. Name some things that require dry heat sterilization. So dry heat sterilization can be used to sterilize things such as paper, powders, petroleum-based products, and sharp metal instruments. Listed below, I have a chart to show you how long you need to autoclave versus how long you would need to dry heat sterilize something. Notice that at the same temperature, 121 degrees Celsius, which is what we use autoclave at, with dry heat, you have to leave it in there for 600 minutes, which is several hours. Incineration is the ultimate means of sterilization. It is a form of dry heat. This slide here shows different temperatures that can be achieved by burning candles, propane, the Bunsen burners that we use in micro labs, the incinerator. Notice the incinerator, the center of it can reach up to 800 degrees Celsius, which is very hot. I used to use this because we used the Bunsen burner that used natural gas. Well, natural gas has different temperatures based on the color of the flame. So number 4, the image 4, that blue flame is very, very hot. And you would want to put your inoculating loop right in the cone, the top of the blue cone that's inside. And if you put it there within a second or two, it turns bright red. Versus over to the far right, left, number 1, if you put your inoculating loop in there, you have to be careful where you put it. You want it at the top of that cone where it's much hotter. This is me using a Bunsen burner, and within a couple of seconds it gets bright red. So we talked about heat extensively, and heat can be separated into dry heat and moist heat. Dry heat, no water involved. Moist heat, water is involved. Well, here's a variation of moist. So now we have heat plus a chemical vapor, and this can be used in things such as the Harvey chemiclave. This apparatus is seen at a dental office. It generates an alcohol and formaldehyde vapor at 15 psi and 132 degrees Celsius for 10 minutes, and it sterilizes everything.